Hi everyone, Professor Gustin here. I'm making this video to give you an appreciation for what's on Lab Practical 1. A lot of times students are kind of confused about how do I do an online lab practical or even lab practicals in general, whether they're online or live. So I'm just going to kind of give you some pointers to help you perform at your peak for Lab Practical 1. Lab Practical 1 is on labs 1, 2, 3, and 4. And right in front of you right now, I have the lab book. And I'm just going to run through here and just give you some pointers. It doesn't mean if I don't say it, it's not on there. But I'm just trying to emphasize to give you an appreciation for the kinds of things that are on there. Everything on the lab practical is somehow in the lab handout. So use your lab handouts. Hopefully they're completely filled out after you did each lab. The kinds of things. Here I have a whole list of routine safety practices. I might ask you, tell me some things that you would do that are routine safety practices. Make sure you recognize what these are and be able to, point out some of the most important ones. When I put questions in the lab handout, like right here, a lot of times I ended up putting these questions in because I found students didn't know this, despite the fact that I gave this introduction thing. I find that students didn't pick up on these points. So when I write questions in here for you to fill in, they specifically are things that I'm likely to answer. I'm likely to ask on a lab practical because I really want you to know that. It doesn't mean I don't want you to know everything else, but for certain, make sure you know these things that I've asked you to write out. So look over those. All these things, safety routines you should perform before leaving the laboratory. Make sure you know what they are. You can see some of the virtual labs, I don't have any questions after. I have nothing to write here. And so what that's telling you is this was kind of reinforcing that idea, but there's nothing in particular that I'm going to pick out of here that's not on another question. Again, here's just asking you a little bit about hand washing, and I ask you to summarize it. So I would definitely commit that summary to memory. What is the recommended method for hand washing, and what organization recommended that? Where would you go to find that? Right here, you did a little video, and then you had to answer some questions about the video. Now, these answers they don't have the questions here, they just have the answers. So I'm not going to ask you to memorize answers to questions that are not written here. But I do want you to be able to distinguish between biosafety level 1, 2, 3, and 4, or at least have a general understanding of the differences. Here's another thing, standard practices. That would be something that you could remember, standard microbiological practices. This is the kind of thing I might ask you. I give you an example of a situation and you tell me, is it biosafety level 1, 2, 3, or 4? Here's a bunch of virtual labs, and we did some metric measurements. And then I put some questions here. And so right here, I asked you these three because I want you to remember those. Anybody in a scientific field should understand at least the basics of temperature in Celsius, what's freezing in Celsius, what's boiling, and what's body temperature. So you should memorize those numbers. These questions right here, they're asking you to calculate, what's the metric unit for volume? How do you calculate milliliters to liters? I'm not necessarily going to give you these same numbers, but I'm very likely to ask you to do something like that. Or I might ask you to calculate, I might give you, 2 liters. How many milliliters is that? So make sure you can go back and forth between the units in the metric system. What's it mean to terabounce? So vocabulary shows up a lot on the practical. The basic measurement for weight, and convert between milligrams and grams, or convert between units of the units, metric units of weight. So these are the examples of kinds of things I could ask you on the lab practical. I probably won't give you the identical numbers. In lab 2, it talks a lot about the microscope, and that supposed to be one word? There we go. Let's see. So one of the things I do is it gives you, oh, we did a lab assignment. And so I put some questions here that you should remember. So when you wrote these out, these are things that I wanted you to remember from that. I give you a lot of vocabulary related to the microscope. So I would recognize those vocabulary words. And a lot of them are parts of the microscope. Some of them are, well, a field of view, the circle of light. That's not really a part of the microscope, but that's created by the microscope. So make sure you know what all these things are. So know the vocabulary. Here's a picture of a microscope. You should be able to point to those parts on this microscope and give a general description of what they do. Again, right here, this lab doesn't have any question after it, but notice down here I do ask you a question. With what objectives would you use oil? So maybe from that lab, you would be able to answer that question. So these questions I put here are so you have something to study for the practical. So do not ignore questions that I've asked you to write them out. When you do total magnification, here's something you would have filled out from the lab handout. And then here it's asking you, I think somewhere, it asks you about total, oh, here's one that says, let's see, well, I can't remember where the question was, but you should be able to calculate total magnification. If I told you the magnification of the oculars and the objectives, you should be able to calculate total magnification. Here's a function of parts, two parts that can adjust light intensity. So that's on the previous section talking about parts. Distinguish between prokaryotic and eukaryotic cells. And I think right here it gives you an example of some of those kind of questions I could ask you. These would be really typical questions you would see on the lab practical. I might give you a picture and ask you something about that. So let's see. Yeah, like I might, in here, I might, let's see, name three parts, prokaryotic cells, and label the prokaryotic cells. 3 parts of cell. Yeah, anyway. So I might give you a picture like this and ask you, similar kinds of questions. That's really typical on a lab practical. There's going to be a lot of pictures. And again, make sure you know the difference of prokaryotes and eukaryotes, even just by their names. Here we have lab 3, and it's talking about preparing specimens for the microscope. Again, I ask you to do these summaries because that's what I want you to remember. Okay, here's a bunch of vocabulary words, and you're going to match them. You should be able to match them to a definition. Smears and simple stains. Whenever you use a word, smears and simple stains, you should know what those are. Why do I do a smear? Why do I do a simple stain? Actually, that's a big thing in a lot of the labs is the why. Why do we make simple stains in microbiology? Why do we make smears? And so the other one is make sure you know the names of the tools. What is this thing, this inoculating loop and a Bunsen burner? Okay, so there's a lot of pictures, or there's several pictures of tools. Make sure you can name them. They don't all have matching answers. Some of them you actually have to see this and name it from memory. It might be fill in the blank. There's a simple stain. It's asking you, why do we use a simple stain? So make sure you know answers to that. We do a bunch of different stains in this lab, on this lab practical, and you want to know why you do each one. Why don't we just do simple stains for all of them? Or okay. Oh, here we go. This is the question I was looking for. It says name common shapes and or arrangements in the cheek smear, the cheek cell smear. I'm not going to ask you to remember the common shapes and arrangements of bacteria of something you did in lab. What I would do is I would show you this picture again, and I would ask you that same picture. I'll show you a different picture and say, what are the shapes and arrangements of these bacterial cells? So you want to get your Coccus bacillus spiral, Staphylococcus, Streptococcus, Diplococcus, all that straightened out. Okay. Here, again, shapes and arrangements of bacteria. There's a bunch of streptococcus, and I'll give you this picture in your lab, on the lab practical. Again, it might not be this picture, it might be a different picture, but be able to answer questions like this about similar pictures. Wet mounts. Make sure you know why we do a wet mount. What's it to be modal and non-modal? Vocabulary. What's Brownian movement? So vocabulary is important. So this is a lot. Again, these questions come from things that I've put on lab practicals in the past, and students have gotten wrong. Not enough people understand what a smear is, or what does it mean, what's lens paper for. So I put these questions in here. So definitely make sure you know the answers to those when you go to do the lab practical. And again, it's not all going to be multiple choice. There's a lot of fill in the blank. Blotting paper, bibulous paper. Make sure you know shapes and arrangements. You did this in lecture as well as lab, so you should be able to tell, oh, these are coccus and they're in clusters, so they're staphylococcus. These are bacillus and they're in chains, so they're streptobacillus, that kind of thing. And so you'll see pictures like these on your lab practical. Oh, there's all the answers. And then I think we have lab 4 is the last one. This is getting into some different kinds of stains, so make sure you understand this vocabulary. Again, I put blanks here because I want you to remember this. What's a differential stain? We're going to do a bunch of stains in this lab. Notice it's important to see the colors. With this gram stain, the red versus the purple, it's a big difference. So when you're studying these, make sure you're looking at these in color. With the endospore stain, the green and the red is important. So you're going to look at those, make sure you look at those in color when you're studying those. We talk about the gram stain technique. Make sure you understand it's one of the foundational techniques in microbiology. So make sure you understand why we do a gram stain and how we do a gram stain. You see there's a lot of vocabulary here, and I have a lot of blanks to fill in. Make sure you understand the concept of a gram stain. What happens, what does each step do? How is each step important? You should be able to name the reagents and why, what happens with that reagent, and what happens when you're a gram positive or a gram negative cell. So here's some practice. And again, things like this could show up on the lab practical. There's some more, and helping you understand why it's important. So you don't have to memorize that strep pneumoniae, whether it's gram positive or gram negative. But if I gave you a picture of it, or I gave you information about it, you should be able to fill this in. Okay, so you don't have to memorize these. You just have to be able to do something like this on the lab practical. Endospores can be a little bit challenging, but look at the color pictures of endospores. Make sure you know why cells make endospores. Look at this, how they, an endospore can germinate, make a vegetative cell, and they can continue to reproduce. Under harsh conditions, the endospore starts forming in the cell, and eventually the vegetative cell disintegrates and you have an endospore. So make sure you can understand that diagram and the importance of endospores. And there's endospore stain. Let's see, somewhere we talked about 2 different kinds of bacteria. Oh, here it is. The 2 most common medically important genera of bacteria that make endospores. You got that in lecture. And you should be able to answer that question. Acid-fast staining. Why do we do acid-fast staining? To find bacteria that make tuberculosis, that cause tuberculosis. So know a little bit about acid-fast staining. If you're in the hospital and they have, your patient has had a sputum sample and they did an acid-fast stain on it, you should know that they suspect tuberculosis for that patient, or at least they're trying to rule it out. Capsule stainings. You talked a little bit about capsules in lecture. Make sure you understand why do bacteria have capsules, and recognize the capsule stain. Where is it? Way up here. There it is. This one with the unstained carbohydrate-based capsule around the cells. So Make sure you can distinguish among these different kinds of stains. I think we had flagella stain in there too. Talked a little bit about that. That's it. That's what's on the lab practical. Everything you need to study should be in your handouts, okay? If you have any more questions about it, please reach out and study. This is not something, you're not going to use your book. You're going to need to really study for this. So, and it's 50 points, so it's worth a lot. Good luck.